Hi, ma'am. Welcome to Teach Nook. As a mentor and as a trainer, we welcome you, Rachana, ma'am. So are the candidates, Purva and Manasa, who is going to be a student for whom you are going to take the classes for microbiology. So you can
Okay,
start
sir.
your session successfully, ma'am.
Okay, thank you. Thank you so much, sir.
Thank you. Have a good day, ma'am.
I hope I'm audible.
Yeah, ma'am, you are audible to me. I think so. For everyone, it is audible. Guys,
Okay.
Yes, ma'am, you're audible.
Okay, thank you. Kids, I just need one thing from you people. If at any point I get my voice gets distorted or you aren't able to hear me properly or you won't be able to see the content, kindly let me know, okay? Do not hesitate about it. So, as sir has already told you my name, I'm Rachna Tiwari and I'm going to take take your classes on microbiology, okay? Let me share PPT. Today we are going to start with basic microbiology, okay? Before going or moving towards the major topics, I want to clear and lay out the area, lay out for what basic microbiology and what we are going to deal in it, okay? Um, First of all, please let me know in what classes or in what particular class you people are in. Hello.
Purva and Manasa, just give your introduction. Why every mics are mute? So just go with the mentors only.
Yes.
They can come to know about yourself, guys.
Hello.
Yes.
Hello, am I audible?
Yes, dear. You are audible.
Hi, Rachna. I'm Purva Saxena. I've done bachelor's in microbiome, and right now I'm pursuing master's in microbiome. And
Okay.
yes, and I want to do a dissertation on immunology and diagnostic, and that's the reason I joined um, this.
Classes.
Yes, this class.
And what about Mansa?
Hello, ma'am. I have completed my MSc in biotechnology, and
Okay.
I'm looking for a job, so I have joined this for the job oriented purpose.
Okay. So I guess you people you have done, as you told, that you have done the MSc and the other one is doing the masters. So let's start. Let's begin. And again, at any point you get confused or you don't understand, kindly let me know. Okay. Now I think my screen is visible to everyone.
Yes, ma'am.
Okay.
Yes, it
So
is.
okay. So kids, before starting, I want to say or give a little introduction about microbiology. So, what's the issue? Yes. So, what is microbiology? I guess the word suggests you can bifurcate, just bifurcate this particular word in two different sections. This particular word says microbiology. Micro means small. Okay. Now, small, and by small I mean those particles or those elements which we are not able to see with our naked eye. Those particles or those elements or organisms are considered as micro. Okay. Now, when we talk about microbiology, by biology we mean the living organism, and in this particular section we learn about those particular elements or organisms which aren't visible by our naked eye and are considered as harmful, although they are not completely harmful for us. They are not it's not like they're useless, they are just there to harm us or something. They do have certain properties, and that particular properties we explore while researching about them or utilizing them in the say in production purpose, or say for the production purpose of like medicine in pharmaceutical industry, or say in food industry for like enzyme production and all these things. So what is microbiology? It is the study of those particular organisms. Okay. Now, when I said biology means living organism, we suddenly get confused by then why do we study virus in it? Viruses are also considered as the they are also considered, they are also the microorganisms, and we do study about them in microbiology, kids. So does what makes them fall under this particular category? is, although they are considered as the non-living organism, but whenever they enter the host, whenever they enter inside a particular, say, human being, or say any animal, or say plant, anywhere, wherever they enter inside a host, they shift towards being a living organism. They cannot live by their own because they do not carry their own, working mechanism. Like they do not carry everything with them, so that because of that they are considered as a non-living organism in beginning, or say outside the host. Now I think you can see there are branches which have been listed below of microbiology. I know it's a very small list. There are many, many other branches of microbiology. And one more thing, microbiology is one of the oldest sections of science, or say advanced section of science, because in when we started learning about science, there was only two sections, zoology, botany. Later, when people started exploring, they came about that microorganism, and that's how microbiology came in contact with everyone. Now, in branches, you can see these are particular sections. In pure microbiology, you completely learn about microorganisms and their characteristics, how you can isolate them, how you can characterize them. And by characterization, I mean how the staining process, or say the genome study, all is this comes under pure microbiology. Bacteriology completely deals with the study of bacteria. Mycology completely deals with the study of mycotoxin. Now, what is mycotoxin? Mycotoxin is a study of certain microorganisms are capable of secreting different types of, say liquidy items, and those liquidy items are considered as toxin for as human beings, or say other organisms. Those particular compounds are characterized as mycotoxin. When you study about the mycotoxin, I can name a few examples like aflatoxin, ochratoxin. These are particular mycotoxin. And we study about this particular sector, it is considered as the mycology. These studies sections I have listed below, and we'll study whenever when the topic comes. I'll give the brief introduction about that particular topic, and we'll study in deep. If I go on telling about these particular topics, it may vanish later on. Now, what the microbial world consists of? Okay, now there are organisms, and although living and non-living, two categories has been divided. Okay, uh, in the say one category we keep bacteria, archaea, eukaryotes, and under that eukaryotes, algae, protozoa, fungi, helminths, protists, they come under the eukaryotic family. And one is the prokaryotic family. Prokaryotic, and basic difference between pro and eukaryotes are considered as the true organisms, because they contain their own nucleic acid, their own say nucleus and everything. That's why they are considered as the true organisms. And the prokaryotes are considered as the primitive. These organisms have been in our sustenance from many decades. Okay, and they have evolved, and that's why they are considered as the primitive ones. Next, the infectious agents, or say in the non-living one, we study, or we see there are few organisms which have been listed are the viruses, viroids, and prions. Again, when we'll study about particular topics, say bacteria or virus, we'll deal about it completely. Okay, I hope up till now it's quite easy and feasible kind of thing. It's not
Yes,
something that's beyond our understanding.
yes, it's understood.
Okay. Okay. Now, in the applied sciences, as I told you earlier, there are many more sections of microbiology. So in microbiology, when when we broaden our spectrum, okay, when we broaden our spectrum, we tend to move and merge with different sections of society, or say with different sections of sectors of society. And in those sectors, microbiology has been indulged or have been added with these particular sections like agriculture microbiology, food microbiology, microbial biotechnology, industrial, pharmaceutical, and medical. Now, The overall, you can say the summarized answer for all of this is microbiology, or say the study, or these microorganisms are being applied in these particular sectors for only one sake, that is for betterment. Like for if production of, I'm doing a production of antibiotics, say I'm producing some sort of vaccine out of a microorganism, that will be considered under medical microbiology. If I'm just implementing these microorganism, or say gene of these microorganism, or by any mean for the betterment of, so that I can create some different kind of crop which are resistant towards bacteria or these microorganisms, those, that particular field deals with agriculture microbiology. With the same particular logic, if say I study microorganism, I do some kind of gene cloning, or say any kind of I create a difference in microbes, so that I can reduce the percentage of food spoilage. I can reduce, or say I can increase the percentage of food sustenance, the shelf life of a food material. That particular study we study under food microbiology. Next, also microbial biotechnology. Microbial biotechnology is one of the booming subjects. So I'm not saying this other subjects are not booming. I'm saying microbial biotechnology is booming because it's kind of a newest compared to the other subjects. Microbial biotechnology is a complete merger of two grand and two complete, say, larger sectors of our science field, and that's microorganism and biotechnology. And again, in this particular sector, we study completely about the DNA, RNA, its say genome, its gene, its alleles, not alleles, um, genome sequencing, say DNA sequencing, markers, and everything we study under, and that too in the respect of microorganisms. Complete implementation of biotechnology, the complete implementation of technology, so that we can study and understand microbes that we deal under microbial biotechnology. Now, I guess I left one or two. Industrial microbiology. In industrial microbiology, kids, we learn about how we can use these microbes to create some kind of product. Now, what kind of product I'm talking about? I guess I used a word before, enzyme production, right? So enzyme production is a kind of industrial product. Enzyme production is very useful. I think you might have seen a kind of advertisement in your say home or while watching television or something. There's a product, an enzyme which has been advertised very widely and at a large scale, called as Yakult. It's a digestive enzyme that we came to know about, and that kind of enzyme helps people, particularly who are suffering from some kind of digestive issues. Okay, so certain enzymes, like say citric acid production we do, say alcohol production we do, these all particular things can be produced with the help of enzymes. Alcohol production, acetone production, say cheese production, that yogurt production which we do in our home, say on daily day-to-day life. Enzyme production, I've already told. Textile production is also a sector of where we use microorganisms for say any kind of textile production. So these all categories, or these all small examples, these merger, or these kind of things we study under industrial microbiology. Now, next, which we are going to deal about is the microbial diversity. Now, before moving towards microbial diversity, I just want to know from you people, do you people comprehend or understand this particular word diversity? Am I audible?
Yes, you are audible. It
Yes.
means like the microorganisms are all over present in the environment or in the atmosphere, and the relationship between the environment and the particular with the microorganisms.
Yes, kiddo, you are right. Uh, when we simply learn about diversity, diversity means the level of species or say strain of anything you can find in your nearby area or say in the environment. Uh, the exposure or what's the correct word for that? Number of different kind of bacteria are there, right? Different kind of viruses are there, different kinds of plants and animals are there. So this particular thing where we study about anything, say any kind of species you are studying and you're studying how much type of that particular species is present in an area, and its relationship, like you just said, its relationship with the environment. This particular thing is considered as a diversity. And when we think or when we study this particular thing with one particular perception in our mind, that is microorganisms, and when we try and understand the kind of microorganisms are present in any particular area, is known as microbial diversity. Now, I guess you people have learned about a particular there's a very simple experiment we do in our microbiology lab, and that is known as serial dilution. I guess you people have performed it in your laboratories, or you must have learned about it maybe.
Yes.
Are you familiar with this
Yes,
particular term, serial
Yes. Yes.
dilution? Yes.
yes, ma'am.
So this particular thing where we do serial dilution, what we do is we try to isolate a single type of bacteria or say microorganism from a very diverse um, say stock solution. Okay, say we took in serial dilution we mostly take 1 gram of soil sample and we just keep on diluting it. Okay, from say if I'm diluting it at 10 folds or say 10 times, I'm diluting it. What I'm trying to do is I'm trying to isolate a single type of bacteria from a colony or say multiple type of microorganism which I have extracted, which I have accumulated. The correct word will be accumulated, which I have accumulated from a simple soil solution from whatever area you're trying to understand. When we talk about microbial diversity, what are the things that diverted a person what are the things that diverts a person towards this particular sector, microbial diversity? Microbial diversity is a very word. What I can say is very good for our research point. Okay, now how it's good for, because uh, the only thing or the only reason because behind doing any kind of research is motivating or you know trying to understand what kind of microbes are there in the environment and what are the benefits that we can accumulate or we can get from them. So if I understand say I have a single particular I'm taking a single type of bacteria here, say coliform bacteria. Okay, now coliform bacteria is a very diverse category. In this particular, it comes under that gram negative bacterial species, and it contains all kinds of species diversity, say like Staphylococcus comes under them, E. coli comes under this category. So if I study coliform bacteria, I can understand a basic outline of what kind of say I just told you 2 3 names. Okay, so if I study coliform bacteria in general, so I can understand what as these bacteria come under them. So it's quite we can so say you can conclude a rough conclusion you can give is maybe these are all they create they give or they cause, but they cause similar kind of disease, although and the diversity or say because of diversity that we cannot simply say that all are same. But understanding a single kind of bacteria under a category gives a brief idea about what others can be like. Okay, now under microbial diversity, the first thing that not the first thing, what I'm going to discuss you with is discuss with you people bacteriology. Now, what is bacteriology? As I told you earlier, bacteriology is the study of bacteria. Okay, not only the study of bacteria, it's complete study of Bacteria, by complete study, I mean it's the capsid, it's surface antigens, say the nucleic acid it contains in them, kind of organelles it contains in them, what kind of toxin or say antibiotic it can produce, what kind of enzyme or say industrial product I can build from it. This complete study I can gather. I know I just distribute or say I separated these industrial things with the industrial microbiology, but in the grand total, these all these studies all comes under bacteriology. Now, the important species in bacteriology, it contains only 4 5 species in this. It's not like not like that other microorganisms are not good for us or say they aren't important. I've just tried to create give a rough idea of these species. Now, Staphylococcus aureus, Pseudomonas aeruginosa, Salmonella typhi, Escherichia coli, and Clostridium tetani. I think is some of these bacteria you have learned in your graduation or say post graduation about. Uh, there's a bacteria present in our gut. I hope you people are familiar with it. Hello.
Yes, ma'am.
Okay, I'm just trying to ask here, kiddo. There's a bacteria present in our gut. Do you know what kind of bacteria is present in our gut?
Pseudomonas.
No, love, not that's not Pseudomonas.
I-
It's Escherichia coli.
E.
E.
coli.
coli is already E. coli is present in our gut. It helps us with digestion. It helps us in proper digestion of food, and and a very limited amount of E. coli is present in our gut. But it does not mean that E. coli is good for us. If by any mean, E. coli is mostly present in that dirty kind of water, say dump water, the water which is still it's not moving. That kind of water sources contain E. coli, and E. coli like ponds and lakes which are does not get cleaned now every now and then. Those usually generate E. coli in them, and by any mean, if a person ingests E. coli, and that person will start getting it will get diseased from that particular microorganism. Common type of symptoms which a person from infected from E. coli can get. Simple symptoms are like stomachache, fever, chills. After that, extreme, you can say the temperature starts increasing. Say it will increase from 100 to 104, and it will keep on increasing. Hallucinations, these kind of things, a person gets if get person gets infected from E. coli. The normal water, drinkable water, we drink water. Some get their water from say directly from the tap or say directly from say use RN, we use RO. But there's a standard for Escherichia coli or say coliform bacteria. uh, Drinkable water must not exceed 50 mpn in when we are talking about coliform bacteria or say E. coli. It's a standard unit. A drinkable water must not exceed 50. And by 50, I'm saying most probable number. Okay, mpn by there's a experiment known as most probable number in which we calculate the percentage of coliform bacteria. I think you people have done this particular experiment in your again graduation or post graduation. Now, when I'm talking about these particular diseases or say Escherichia coli, Salmonella typhi is responsible for typhoid, and Clostridium tetani is responsible for the tetanus. Staphylococcus aureus, it's responsible for skin infection and respiratory infection. Same goes with Pseudomonas aeruginosa. It also gives both types of infection, that is skin and respiratory issue. Now, habitat of bacteria. Now, bacteria are not specifically aerobic or anaerobic. I think not. I don't. I shouldn't use this word like I think. We we generally study about the first few years of biology, or say in first few years of microbiology. The common thing which we run around is aerobic and anaerobic. Now, what is the basic difference of aerobic and anaerobic? One who can live in the presence of oxygen, and who can live in the absence of oxygen. This is the basic difference with aerobic and anaerobic. Bacteria does not fall under a particular category. They do not follow or fall under aerobic or especially the anaerobic criteria. They follow, or they are widely distributed in both the categories. Now, you can find bacteria in soil, water, air, everywhere. Uh, we do get in contact with different kinds of bacteria in daily life, but uh, it's not like that we get infected. Say I get infected from rhinovirus responsible for the common cold. I got infected today, and it's not like that. After I get healed, that bacteria vanishes from the environment. They are there. They are always there. But whenever a person gets infected, the body starts producing antibody. I mean, it's in immunology. I guess you people have learned about it. So the body starts producing antibody, and that antibody protects us until the next infection, I can say. So bacteria are always present in environment, and by this I do not mean that all kind of bacteria are always present in the environment. There is, that's why we use this particular word, microbial diversity. In particular sector, some different type of bacteria are always found. That's why it's been seen, or it's been studied that some particular sectors always get infected by some or other type of or similar type of disease every now and then. Now, uh, uh, again, I'm talking about the habitat of bacteria. As I told you, our body also contains E. coli, right? It's present in the gut region. So human body also contains a considerable amount of bacteria in our cells. By human body, like in gut also, it's present in our skin. It's present in our the reproductive areas also. Bacteria, particular say amount of bacteria are always present in our mouth also. Our mouth also contains a particular type of bacteria every time. Again, the mouth bacteria helps in the digestion process and breaking down of particular food component. Now, uh, there are as different as temperature differs from place to place, right? So the bacteria who can sustain hot temperature, or say hot springs temperature, if they can survive in the hotter areas, those particular bacteria are considered as the extremophiles. Okay. By extremophile, again, I'm going to repeat the same thing. If a bacteria can survive in a hotter spring or say hotter area, those bacteria are mostly considered as the extremophiles. Now, there's a very good example of an extremophile I can give. Um, Milk contains a particular type of bacteria. Do you people know what kind of bacteria is found in milk? Hello?
Lactobacillus.
Yes, thank you so much. Now, lactobacillus, and we never drink the raw milk, right? People do drink raw milk, but should not be done because it contains multiple type of microorganism, which is not good for our gut region, or say our stomach or digestive system. Although, the lactobacillus, whenever we boil the milk, lactobacillus does not die. It never dies. It can attain, it can sustain, it can resist hotter temperature, or say the boiling temperature we are giving to a particular bacteria. So they can be, or they are mostly considered as the extremophiles. Okay. Now, next one I'm going to discuss with you people. Okay, before moving forward, I didn't ask you people. I just kept on speaking. Are you people getting what I'm trying to explain here, or am I going fast, or should I change the type of explanation I'm giving?
Yes, yes. It's very. I'm getting your point, and I understand
You're
everything.
understanding?
Whatever you.
Mm,
Yes.
I can
Okay.
Thought
understand
as
it.
of now. Yes.
Okay. Listen, and yeah, to both of you, I'm just asking this. Just not, do not hesitate. Okay, if whenever you feel like you're getting stuck, you're not understanding anything, so because the basic thing what we are trying to accomplish here is giving you some sort of knowledge about this particular subject. Okay, not some sort of knowledge, but basic knowledge about microbiology. Okay, so
Yes.
please feel free to tell me whenever you get stuck or whenever you don't find what, if you don't understand what I'm trying to teach. Okay, to both of
Okay.
you. Okay, now
Yes.
the next thing which I'm going to discuss is one of the most notorious sorry, yes, this thing we are going to discuss is one of the most notorious microorganisms you can find in the environment. Why am I saying this as a notorious? Because these particular breed are considered as a non-living. Now they're considered as non-living when they're outside host. Again, whenever they enter the host, they not only get changed into a living organism, they start manipulating the entire cell system of our body. And by cell system, I mean if they attack the liver, they'll start growing and degrading. And um, this is these particular organisms. Now, what? Sorry, these particular organisms are also considered as a bigger threat because killing them or destroying is a bit of a sore topic when it comes about killing a virus or destroying the particular virus. It's not like all viruses are like they can't be destroyed, but there are a few which are like a headache. Now, whenever we are talking about viruses, as you can see, there are few pictures in front of you. Okay, now viruses do change, or say they differentiate themselves with in comparison to bacteria or fungi in some say particular sectors. In viruses, there are two kind of viruses you can find in the environment. One is are considered as a naked, and the other ones are not. Now, what is the basic difference between naked and say complete virus? Naked viruses do not contain an outer shell, do not contain outer envelope. Okay, those viruses who contain an outer envelope are considered as the complete viruses, and those viruses which do not contain an outer envelope are considered as the naked virus. Now, just a moment, sir. See, I guess you can see this particular virus. This particular virus structure is like this. This particular virus, sorry, I'm not drawing it properly. This particular structure is known as the icosahedral structure. Now, what I've tried drawing here is this particular type of structure, or this particular type of outer layer is known as the envelope. Okay, and those viruses which contain these envelopes are considered as the complete viruses. Now, if the particular microorganism, say, I'll give you an example of this complete virus, although an example has been given below. Adenovirus is a type of enveloped virus. Now, another example is retrovirus, responsible for HIV/AIDS. Now, apart from this, you can see some sort of antennas which are protruding outside from this particular envelope. These antennas are considered as the glycoprotein, or say not the glycoprotein. Glycoprotein won't be a good word for this. These antennas are considered as the surface antigen. Okay, they are considered as the surface antigens, and these surface antigens are found outside almost all the types of viruses which are found in the environment. And this particular surface antigen is responsible for it's not particularly they are responsible, but they are also responsible for any kind of infection that causes inside a human body. Apart from genome, I'm going to repeat again, apart from genome or the gene or the DNA present inside that particular pathogen, these surface antigens are also responsible for triggering. I'm going to write here, surface antigens are responsible for triggering our and triggering our sorry triggering our immune system. They trigger our immune system, they damage our body by attaching themselves to any kind of cell they are attacking. One more thing. Okay, sorry. One more thing. After this, these surface antigens, I've told you about the surface antigens, right? Now, these surface antigens are responsible for the different kind of strains too. Viruses tend towards not viruses only, every kind of microorganism, or say in fact human beings also. We uh, whenever a kind of environment changes, say if you are living in a area which has a moderate kind of temperature throughout the year, and a moderate amount of rainfall when the rainy season comes, and you had a tendency of living in that particular area, say for 10 to 20 years straight. Now, by because of any reason, if you change your locality and you move towards, say, you move towards a place which is hotter or say humid as compared to where you are living right now. For first few days, for first few days, our body will react. It will give all kinds of tantrums. But after few days, for first few days we have to will get all kinds of different kinds of problem, we'll face problems. Okay, now after few days what will happen, our body will start adjusting. Okay, now by adjusting I mean our body develops a mechanism, a coping mechanism, uh, where or by which we start we do not get all kinds of our body do not throws all kinds of tantrums, or we do not get irritated by that environment change or the locality change. Now this happens with microorganisms too. Now what happens whenever bacteria or viruses they change or they I'm going to just talk about virus right now, we'll discuss bacteria too later. Now what happens, and I have an example too. As you can see, there's a virus, or say we are facing from past 2 3 years, right? COVID virus, SARS-CoV-2 virus. Okay, now COVID virus was previously came in existence in the year 2012. Now after few years we started facing this particular disease again. In 2012 the death rate was not that high, it was really low, or say people faced only few symptoms or few diseases, few problems. Respiratory issue, they did have respiratory issues, they did got fever, but it was under control, and it got eradicated easily. But later on this particular virus changed, it got evolved, and by evolving I mean the COVID virus is continuously evolving by just one thing. The COVID virus and all kind of strains which we have seen in past few years, the one thing which has been considerably similar in everyone is the surface antigen gets changed or gets modified or gets there's always a say um, some sort of similarity from the previous one and some sort of difference from the previous strains. The surface antigen specifically gets changed, and by that change of surface antigen, this particular virus, say when I'm talking about this particular virus, I'm saying COVID virus, so or the strain of the COVID virus also changes. Now, what are surface antigens? Now, surface antigens are mostly a complex molecule made up of glycoprotein. I think you people know what do you people know about you've learned about glycoprotein? Hello, hello,
Yeah, ma'am, it's
Hello.
audible.
hello,
Yeah, it's audible,
hello.
ma'am.
I'm so sorry. I thought they got disconnected.
No, no,
Have
no,
you
no.
had you people learned about glycoprotein in your say when you were studying?
Yes.
Yes,
Okay.
ma'am.
So you people know what glycoprotein means. It's a mere combination of carbohydrate and protein. So surface antigens are also made up of, or these surface antigens are nothing but glycoproteins. It can be a glycoprotein. It can be a lipoprotein. Sorry for this horrible handwriting. Oh, no, this is, I guess, better. Lipoprotein, um, glycoprotein,
Protein.
yes, the glycoprotein. So it can be a glycoprotein, it can be a lipoprotein, it can be a lipopolysaccharide, which I'm going to write here. Lipopolysaccharide. There's a when we are talking about lipopolysaccharide or lipoprotein, everything. Have you people ever noticed that whenever we give this name, we name it in a very different way? Here, glyco word is small, protein word is completely written. Here, the lipo word is small, but the carbohydrate word is completely written. Have you ever noticed we always name them in this funny way? Why?
No.
That's because, kiddo, the naming was done with a very with a very the person who came up with this idea was very intelligent. I'm saying because in this particular word you can completely understand the percentage of these two compounds. In lipoprotein, the word lipo is small, so it only contains 30% of lipid is present, and as a protein word is completely written, the percentage of this compound in a lipoprotein will
70%.
be, say, around 70%, but it will be higher as compared to lipo. The same concept goes with glyco and protein also. The concentration of glyco is less, say glycogen or carbohydrate is less and protein is higher. Same
High.
thing goes in lipopolysaccharide also. The percentage of protein polysaccharide is higher
30%.
as compared to lipo. That's why we came up with this interesting way of writing the name, so that you may not guess the exact ratio, but you will get the gist and of how or what is the percentage of these two compounds in whatever thing you are studying about. So after as sorry, I got off script. We were talking about envelope. Now after envelope, inside an envelope, if I'm drawing this right, see, this is envelope, this one is envelope. Inside, this is a bacteriophage. Bacteriophage is a virus which infects bacteria, right? Now, inside an envelope, I'm just going to mark it as E. Inside an envelope, there's a another layer. Just a moment. Yes. There's a layer of protein. Okay, there's a layer of protein which is known as capsid, and capsid is uh, one of the biggest defense mechanism of a particular virus because it does not get broken easily. It cannot get you cannot destroy or disintegrate capsid by mere say an antibiotic or say with the help of any simple kind of medicine. It requires certain work to destroy a capsid. Now, inside a capsid, when you're talking about capsid, inside a capsid, okay, sorry, one thing I forgot about capsid. Capsid is made up of capsomeres. Now, what are capsomeres? Capsomeres are protein molecule which adjust or which makes or turns them into a circular shape and arrange themselves in this manner and make a hollow capsule-like structure. And that capsule-like structure is called as the capsid. You people have may have seen a capsule or that medicine we get, a capsulated medicine we get in the same say um, structure. Hollow capsule gets formed with the help of capsomeres. So we were top. Sorry, so sorry. No, that's better. Now, inside is a capsid present, and inside that capsid the genomic material. It can be single stranded or double stranded. Okay, single stranded or double stranded, and by double stranded I mean it can be a single stranded DNA, it can be a double stranded DNA. Likewise, it can be a single stranded RNA. Sorry, it can be a single stranded RNA or a double stranded RNA. Now, and when we talk about a genomic material, specifically RNA of a virus, they also make a category or say like some sort of it gets bifurcated into two categories. It can be negative stranded, it can be positive stranded, and this negative and positive stranded are mostly called as positive sense RNA or say negative sense
Since RNA.
RNA. They get bifurcated into different categories. That's why viruses are considered as one of the notorious things. They carry all different kinds of things, and every kind of strain is different from the last one. Now, this is a an outline of all the things you can see inside a virus. I think I was able to tell you the outer layer, a brief explanation of virus. Did you people understood?
Yes, understood.
Yes, ma'am.
So,
Ma'am, I had one
these
small question.
yes, dear.
What is envelope made of? Like, is it made of protein or carbohydrate?
Now, the envelope is made up of all three things. These are made of, or not all three things. Mostly, it is made up of carbohydrate and lipid. Mostly, it is made up of carbohydrate and lipid. Sometimes, say few percentage of protein is also present in the outer layer or say in the envelope. But whenever we talk about capsid, it is completely or solely made up of protein and protein only. Okay,
Okay.
now where were we? Yes, we were talking about, now we're going to see some of the common viruses and kind of effect we see. Now, papilloma virus, it's a particular type of virus which affects or infects the reproductive organs of a human being, and it mostly causes the genital infection or say cervical cancer of kind of things. Adenovirus is one of the um, most studied virus in past few years. Why I'm saying this because you people are familiar with Covishield and Covaxin. Those vaccinations were given in were made in labs in India.
Yes.
Okay, and Covishield was made. You know, just before we start, vaccines are vaccines are made up of. In vaccines, we remove the genetic material. The gene or the genome is removed. And mostly it is when we remove the gene or genome, it's only the outer layer which gets injected inside a person's body. For the vaccination purpose of Covishield, adenovirus strain was used. COVID strain was not used for creating Covishield. Whenever we create a kind of vaccine against a particular disease, we tend, or the researchers tend towards using that particular strain only. Okay, I'm going to repeat again. Whenever vaccine is made, that particular organism is used for creating that vaccine. So in this particular, if we go this with this theory, the theory says that we have to create COVID vaccine with the help of SARS-CoV-2 virus. But at that particular time period, this particular virus wasn't studied completely. And for for the just for the sake of saving people, we had to make a vaccine. And Covishield was made using adenovirus strain was used for creating this particular vaccine. Now, adenovirus, what this particular disease, why this particular organism was used. As you can see, I think it's written in front of you. It also causes respiratory and gastrointestinal disease. The person whomsoever got infected with COVID or say SARS-CoV-2 virus, they also showed same kind of symptoms. They had respiratory issues, extreme respiratory issue. They were coughing up. Some were when got infected, were infected from many days. Infected. They were coughing up blood. The respiratory tract was completely inflamed, and they were also suffering from diarrhea or some sort of gastrointestinal disease. That's why adenovirus was used. Another reason for using adenovirus was coronavirus. The outer structure of coronavirus somewhat, not completely, but somewhat resemble the outer structure of adenovirus. That's why that also gave the idea of creating a vaccine using adenovirus. Now, the next kind of virus written here is reovirus. Viruses also cause respiratory and gastrointestinal diseases. Okay, whenever we are dealing with say microorganism, there's a kind of symptom which precedes it, and that symptom is known as bacteremia. Now, bacteremia is nothing but when the concentration of microorganism increases in a particular organ or say cell, it starts leaking inside our blood system. And when the concentration gets increased in the blood system, that kind of, mostly it's considered for bacteria only, that condition is known as bacteremia. And bacteremia always comes with any bacterial diseases. Now, when we're talking about adenovirus or reovirus or virus also, it cannot be defined as the bacteremia, but they also show the same kind of symptom, that it also gets released in the blood system, and it ultimately starts infecting the blood system too. Now, next two diseases, calicivirus and the enterovirus, they are responsible for the gastrointestinal disease. Rhinovirus, as I told earlier, rhinovirus is responsible for common cold, the kind of cold we suffer whenever temperature or say atmosphere changes. Hepatitis B virus, it's responsible for the liver infection. Hepatitis B and C both are responsible for liver infection. Smallpox virus responsible for the smallpox. And there's yes, smallpox virus responsible for smallpox. Now, what is the habitat of viruses? As I said many times, they only survive inside any host cell. Okay,
that does not mean that they do not survive outside us. They survive outside it, but they do not flourish outside the host cell, or say the human body. They do not replicate. They cannot replicate. They cannot increase their copy number, but they do survive outside the host cell for replication, for increasing the copy number, for say flourishing. They need a host body. Now, shall I start the fungus as well, or shall we continue from fungus from tomorrow onwards?
Yeah, ma'am, we can start it by tomorrow. Just you can
Okay.
start assigning them some work from what are all the classes you have taken till now. So they
Okay.
can come to have a research about that also. So,
Okay.
and we can schedule the timetable and we can issue it to them. So they will come on the date. So before that, you
Okay.
can give some assignments, what they need to do research or what they need to work on for further days, ma'am.
Okay. One of you kid were telling me that you wanted to do your future research work in immunology. I'm
Yes,
really sorry, I don't
yes,
remember. Could you please repeat your name?
yes. My name is Purva.
Purva.
I
Okay.
want to do
So
your dissertation.
yeah, you want to do your dissertation in immunology, right? So
Yes.
I have a small work for you, resembling
Okay.
your scope, or you're trying to do immunology. Do one thing, kiddo. Immunology basically deals with the antigen, antibody, vaccine production, or say how our body reacts towards any kind of infection, right? So
Yes.
I have a particular project, or say assignment for you. Categorize 5, or say um, take any 5 bacteria, or not, or do both the categories, bacteria and virus both, and make what kind of vaccine is used for that particular disease. Okay. And
Okay.
the disease caused by a particular virus or bacteria, what kind of, say, adenovirus, what kind of vaccine is used against adenovirus, and how that vaccine is prepared. I just need a 2-line answer, that how it gets prepared. It's say it's in the form of toxoid, or say it's in the form of whatever. You'll just get, whenever if you'll search what kind of vaccine is used against adenovirus, the name will pop up directly in Google. Okay. I just
Okay.
need this part. Give me only five bacteria and viruses, and we'll proceed from there. We'll
So
see
I
what
have
can
to
be done from
basically categorize the bacteria and viruses
yes,
and
yes.
their causes and how we can prepare the vaccine.
And you can, if you want, you can search about how you can prepare a vaccine, but for now just give what vaccination, what is the name of vaccination that is used against this particular categories. I just need a name for today. Just find this much and we'll proceed from there. From next I'll try and indulge you in this particular section. It will help you with your immunology research also, and we'll learn more and more about bacteria and viruses. And the
Okay.
other one,
Uh,
Manasa, uh, am I pronouncing your name correctly?
Yes.
Okay, I was afraid I was pronouncing your name. Dear, you have completed your graduation or post graduation?
Yes. Yes. I have completed MSc.
MSc, right?
Now.
Okay, okay.
Yeah.
Then what kind of assignment shall I give you? Do one thing. You prepare. Okay, you do one thing. You give me different kinds of coliform bacteria, and the percentage India-wide percentage of coliform bacteria, where the percentage of coliform bacteria is found more and where it's found less. I just need India-wide. Just go do a Google search and you will find the percentage of coliform bacteria which can found in different regions of India. Okay, I know it's very easy, but I know it's first day. We'll proceed from there. Okay.
Okay.
Ma'am, I want to ask one question.
Yes.
Ma'am, will you also provide me at the end the thesis like project where I have to, because I have to also create a thesis for that particular project on the immunology. So will you help me in that too?
Dear, are you currently working with in project?
Um, Actually, in my college, they told me that you have to do a particular project, and where you have to prepare the thesis for that. So that's the reason
Okay.
I enrolled in this program.
Okay.
So basically, I have to make a thesis on any
Okay.
of the particular project, like related to the immunology.
Related to immunology. So
Yes,
for that, we have to come up with a topic first.
yes,
Okay.
like
Uh,
on a particular project.
okay. Okay. There's no issue. There's no issue. Um, Tomorrow, just remind me. Okay. I tend to forget sometimes.
Yes,
I'm
because
really
I
sorry
have the
for that.
format of the
Just
thesis, but I don't know like how to make, and for that I need your
I
support. Yeah.
understand. I understand, dear. Tomorrow we'll discuss about first we have to come up with a topic, right, on which we have to make the thesis. So tomorrow we'll discuss for say 5-10 minutes we'll take, and we'll discuss what other topics we can work on, and from there we'll start working on the thesis. Okay.
Okay, so do I have to search the topic by myself, or will you help? Will you do this tomorrow?
Okay. Do one thing. It's not like that I cannot help you with this, but little you have to search a little bit about immunology. Why I'm saying you to search, because this will help you write your thesis also. And how it's going to help you is, when you'll search for a topic, you'll try and study about it, and that study we are going to indulge in that thesis only, right? You
Okay,
come up with few topics, I'll come up with few topics, and we'll discuss whatever topic is easable or say feasible for you, and it's also good for your future prospects. We'll work on that. Okay.
okay, okay. Thank you so much, ma'am.
Okay, dear.
Ma'am,
Yeah.
Yes,
ma'am,
Uh,
we will be doing other projects as well, right?
that I think Kevin sir can tell you about another projects or other projects.
Manasa, another project and everything will be shared to you once this 1 month online program has been shut down. Second month, while you're going into offline, there will be a different set of experience and there will be a different projects for you. That will be on complete hands-on experience.
Oh, okay.
Yeah. Thank you. Thank you, ma'am.
Oh,
Have a good day.
thank you too, sir. Thank you,
Thank you,
kids.
ma'am. Okay.
Thank you, ma'am.
May I leave